Hey, AP Bio folks. Couple things today. Now that we have the updates on the AP exam, I'm going to take a little bit of time to go through that, and I'll also be, if you haven't received it already, I'll also be sending a letter from the school with some explanation and some links, and also a letter with the revised scheduling that I'm going to talk about right now. So that should get us set up. Then after we talk about that, so you have that understanding and you get those couple of PDFs I'm going to send, which you can show to your parents, please. Then I'm going to talk about the murder by HIV and use that as our transition to our next technology, which is how do you amplify DNA and then how do you sequence DNA. And we'll start going through that tomorrow. So anyhow, and then later in the week, we're going to, as I assess how AP Classroom went today or yesterday on your quiz, then I'm going to assess how that goes and set up an exam to cover the DNA chapter stuff before biotech and viruses. That exam that we didn't miss, we'll now do on the AP Classroom, and I'll get it set up with a few questions, and we'll get that going and rolling for you. Okay, so let's talk about the updates here. Some of this you may or may not know. Again, it's in the letters that you have or will be getting on email. Anyhow, the date and time for your AP Bio exam this year is now Monday, May 18th at 1 p.m. Central Time. And again, it's a 45-minute exam. And you'll want to be ready before that and stuff, as you'll read in letters and everything, wanting to make sure that 30 minutes of time prior to that exam, you're able to be on, logged in, and ready to go. And all that information about logging in is coming out at a future date. There is a makeup day for some odd reason that you might miss the exam, and it happens on occasion with illness and stuff. That would be Wednesday, June 3rd at 3 p.m. Central. Now, these are assuming you're going to do things at home, open book, open note, open whatever. Now, that's all well and good that you can do that. I will already recommend to you that you be ready to go. I will say it this way. You will certainly, maybe you had in high school, but you will certainly have the opportunity to do open book exams in college. And the minute you hear open book exam, you should cringe because you just don't have the time to be flipping through the book and get real answers. You really have to have the knowledge there and then maybe have the book marked up so you can just quick double check some facts. So that's what I'm going to throw out to you as a word of warning. It is 45 minutes online, 2 free response questions. Question number 1 will be, you get 25 minutes to read and write, and then 5 minutes to upload. That is the larger, that it would be the longer 10-point type free response question. That's going to be 65% of your score. Then you're going to get 15 minutes to read and respond after that has been uploaded. They give you 15 minutes to read and respond to question 2, and then also 5 minutes to upload at the end, and that's 35% of the exam. Okay. As you go on the AP Classroom, and you'll see this in your book or get the link to it on those letters, but I've written it out for you here. Question number 1 is, as I talked about early in the year, and really it's the same thing I've said all the time, that they always have one question like this. The longer one is that experimental design question. So officially by their terminology, and again, you can click the link and go scroll down and see the exact stuff written here in Ms. Franken's letter. Interpreting and evaluating experimental results. You are presented with an authentic scenario accompanied by data in a table and or graph. The sample problem on the link in the AP Classroom course description junk is gives both a table and a graph and a diagram. This question will assess your abilities to do the following: describe and explain biological concepts, processes, or models. And here's the one we've talked about: identify experimental design procedures. That would be controls, independent variables, dependent variables, all those factors that we have talked about and questioned you on before. Of course, analyzing the data and then making and justifying any predictions that you make from that data. And you can see that the larger point values go in identifying controls, variables, etc., attempts, you know, how many attempts, how you're going to do things, and justifying your predictions. Okay, so that'll be that type of question, a longer quote unquote free response question that you'll have time to read, organize your thoughts, and get things going. Question 2 is going to be shorter. It's going to be a 4-point extravaganza. This is the conceptual analysis. When you go to if you go to the links and go to the course description, whatever, it really does, it describes it, but it doesn't have a sample question right now in their stuff. But anyhow, you are again presented with an authentic scenario describing a biological phenomenon with a disruption. The question will assess your abilities to, 1, again, describe the biological concept or process that's going on. Explain that biological, so not just describing, but explaining what's going on. Predicting the cause or effect of the disruption and justifying that prediction. And every part of that is about equal. Okay? So that's the rough, and as I get more, certainly I'll be passing it along to you as more. It also now gives me guidance of types of free response questions to either give you for practice as we continue through this technology last piece, and maybe throw a couple other things your way, and get you prepared. Although I believe right now, since we have done a number of these types of questions all year, and we're not, other than the technology, biotech we're doing right now, I believe you're prepared to do well. We've worked very hard on answering questions properly. All right, so let's talk about our case study. This was a good opening. Again, it was off of a real case study. So as you walk through, and you can look at the case study and read it and walk through the selections, and in each part 1, part 2, whatever, it gave you those scenarios. And in part 1, it kind of explained the background of the relationship the two had, and there's more to it than what they did in the scenario. But basically, it was adding a little application of science to things. If you were on a defense team, of course, you'd be looking for some other infection than the shot that the doctor gave her when he came to his house and called it a B12 vitamin shot, which it was, well, without getting ahead of myself, which it appears to not be. Okay. And of course, if you're on the defense, then you're going to look for any other way that you can get an HIV infection. So if this, you know, as a free response type thing, you're thinking of how many different ways. One, of course, blood donations, but and the second part of that question was, how would you know? How could you narrow down where the initial infection came in? She certainly did blood donations, but those are always tested. You're always tested going into a blood donation to make sure that you don't have any number of infectious diseases or disorders that would eliminate you from being able to donate blood. So there's a number of them, not just HIV. Certainly in her job as a nurse, there would be hazards of any bodily fluids and blood and everything else. Those are always theoretically and should be well documented, especially since the onset or the discovery of HIV retrovirus and stuff, that any of those transmissions, accidental or otherwise, are always recorded. And then, of course, that person is tested on a regular basis to see if they're infected. And then certainly the other way would be sexual contacts. And so they would contact every prior sexual partner that she had had and run them through a test to see where everything was. And that's what the defense would argue. And then it would be the prosecution to track down those things. Certainly the defense would too, and kind of try and throw doubt upon the prosecution's results. Okay. The second part of that then was really kind of, well, how could you figure out that maybe it came from a specific person? Who was the person that could give it to you? And this goes to understanding viruses, that, and especially retroviruses like HIV, have a high genetic variability, a very high mutation rate. Kind of like bacteria, while mutations are rare with things that reproduce rapidly, you can see more changes, quickly. And as you can see, 10 to the 10th virions or viruses per day is the turnover. But here's the thing, again, while mutations are rare and you can have a high mutation rate in things like bacteria and virus and stuff like that, they still have a base genetic ancestry, not unlike, taking a 23andMe test or something like that, or setting up a family tree where you can determine who's, related and stuff. It's the same thing. So any one person infected with HIV could have a number of different variants in their system, developing. And so a number of different variants of the virus there. But if you both had the same ancestral infecting strain, then you can see those variations and trends being similar and trace them back to a common ancestor. Okay. So yes, it mutates and stuff, but two people infected with the same stuff, you can track the evolutionary changes of the genetics, and you should be able to see similarities. And that's where it threw you into the blast, and then you had to walk through the blast. And something we haven't talked about, and unfortunately, we'll probably talk about it after the test, but which is not something necessarily covered on your exam this year, is a phylogenetic tree. That, again, like a family tree or anything else, and hopefully as you went through the instructions and clicked on it, that was the idea, and you would have drawn in there a little tree. And phylogenetic trees, like any type of ancestry, whether it's genetic or comparing animals of different species and where did they diverge and stuff, is a tree that just goes back and traces back common ancestors and stuff. Okay. When did things develop feathers? When did they not? So when you look at those trees, then you are able to trace back to common ancestors and see what's going on. And in this case, the patient, as you read, there was a log of, on the same day that the vitamin B12 shot was given, there was a HIV infected patient who had their blood drawn, except those records were somehow hidden in a box somewhere in, Dr. Well, I actually know his real name from the notes, but Dr. Infectious Buddy, in private files at his house and stuff. Anyhow, So they found those patient records, were able to do basically a phylogenetic tree, and noticed that the ancestry was very similar. And in fact, very recent ancestors that more than any of the other variants had them very close in what was going on. So while the evidence was quote unquote circumstantial, it was quite extensive. And so the jury did find a gentleman guilty, and he was sentenced and stuff. And like I said, it's an actual case. I could give you the names and you can go look it up. If you're interested in a little more background, they had been in a relationship for a number of years. The doctor had impregnated her 4 times. 3 times he had forced her to terminate the pregnancy. The 4th time she said no, and kept the child and raised the child as her own. And then the whole craziness, obviously, he was a little goofy, but then the whole craziness of the B12 shot and the infection occurred. So the idea of putting that out there as a technology piece is just seeing how that you can trace even changes in genetic history back to ancestors and stuff, and use that to do some of that determination, like we said, of paternity, et cetera, et cetera, et cetera. Our next thing, and we'll talk about this tomorrow, is certainly we've talked about DNA and viruses in the initial part of technology. Now it's going to be once you get these samples, a very short, brief discussion on DNA libraries, and then really the important stuff. What's PCR? So if you had even the smallest amount of DNA, how do you replicate it so that you get exact copies? And then how do you use DNA to sequence it so that you know these things? And so we'll talk about gel electrophoresis. And that's the work that, unfortunately, I'll have to film for you, and we'll go from there. So that's really it for today. I'm not going to run through anything else. We'll kind of give you a day to pull back because we've done a lot of work in trying our AP classroom. So after watching this, I don't have an assignment today, but you certainly should go back and start and review the DNA transcription translation operon type of stuff that we did for the exam later in the week. Okay, so that's updates on, oh, and I should say this on the updates of the AP test. If for some chance we are back in school, because in theory we're coming back May 4th, then the expectation is from AP that the exam will occur in the school, even though the format's going to stay the same. You would be taking the exam in the school and not at home. Again, all that update stuff, we'll just keep updating it as it happens. All this technology stuff is very interesting, and hopefully you enjoyed. I think as I've gone through, most of you found the doctor guilty. But anyhow, that's where we're at, and we will have a nice, somewhat relaxing day, and we will talk to you again tomorrow.